Okay, so let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Niao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you, SynBYSS people, for joining us, the 86th seminar, and I call it a University of California, Berkeley Day. I am in Norway after giving a lecture in Germany for a summer school and also giving another talk in Marburg with the Tobias Herb. And we all need breaks and vacations. So I am having a short vacation here until this weekend. And this place is amazing, surrounded by beautiful nature. I love science, but nature cannot be beaten by anything, I guess. Okay, so it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Adam Arkin. He does not need any introduction because we all know him. Professor Arkin is Dean Richard Newton Memorial Chair and Professor at Department of Bioengineering, University of California, Berkeley. He is also Director of the Center for the Utilization of Bioengineering in Space, CEO, CSO of DOE Systems Biology Knowledge Base, as well as a co-director of Enigma SFA. I got to know him in 2006 when SynBYSS, the first SynBYSS center in the world, started with US NSF support. And Adam is also advisor of Chris Boyd, my own advisor. So Adam is my academic grandpa in some sense. And thank you so much for your contribution to scientific community, including co-director of the SynBYSS, and especially all the work related to DOE knowledge base. That's a huge effort and then a lot of benefit to the society, I believe. And the virtual podium is all yours now. Thank you again.
Thank you, Tay. You've known each other a long time, and it's been, you were one of the young folk when we were starting, who were all hoping would become what you have become, which is better than all of us. So it's really fun to be here. It's interesting. I come to these synthetic biology meetings now, and I feel a little bit odd because I've taken such a strong sidebar as I've thought about what I want to engineer. And so I thought I'd tell you guys a little bit about that and see how it goes. We have a short period of time, so I'm going to share my screen, see if it works. You see that, guys?
Yes.
Yes.
Okay, good. So, the big driving force in my laboratory, so very, very early on when I was I guess maybe towards the end of SynBYSS, maybe, I was getting really obsessed with the problem of context, which was the idea that every time I tried to predict how something would work, it wouldn't work as I predicted because the world in which my thing was operating was not what I expected it to be. That it could be something as simple as where the promoter was in the genome, where my cell was in a jar, where that jar was on Earth, and so on. Each of these things had something to do with what happened. And what was really worse than that was that my ambitions weren't to make like a fantastic, valuable chemical product in a jar. My ambitions were to put an organism out into the world and have it do something for me in a way that was trustworthy and beneficial. And that required a degree of trust and a degree of control that nothing I was looking at allowed me to promise. And so I started to become a little bit more obsessed with what are the issues I have to face if I'm going to deal with those problems. And so I thought, well, why don't I think a little bit about where I want to go? And being who I am, I have absolutely no self-control. I thought I'd go for two extreme things here. Let's see if this works. These are two examples that I've framed, I think, where my thinking is. On the left is the diagram that we produced as part of our Center for Utilization of Biological Engineering in Space. And this really is a chemical production thing. It produces food and pharmaceuticals and building materials from an integrated biomanufacturing plant, conceptualized to be able to support 20 astronauts on Mars for about 2.5 years, using nothing but light, atmosphere, water, and if necessary, human waste and mission waste. That's the idea. And what I liked about this was a couple of things, which is that I knew exactly what I was designing for. There was no slop, right? That is, there's 20 people. I had to give them all their calories. I had to, do it from this amount of light and this amount of atmosphere and this amount of water, and I knew exactly what those numbers were. I had very strong constraints. I knew what the costs were, and I knew I had to be competitive with bringing it along. I couldn't impact the planet. It had to be completely sustainable. It was a mathematically constrained, specified object with a known stakeholder, NASA, right, and an understood thing. And what I liked about it as well was that it was forcing me to think of the, how to make things as efficient as possible. So the idea being that I would have to be able to fix carbon and nitrogen without the Haber-Bosch process, without Sabatier or anything like that. I had to be able to use the media and components from one reactor in the other. And I had to be able to process those materials without a lot of crew time, because if an astronaut moves a pinky finger, it costs $1 million. So we got together and began to think about this. I thought it was a really nice way of thinking about, well, what organisms would I have to bring with us? What would they need to be able to operate on? What resources could they have to use? And I liked it because it was a sustainable support of resilient life in extreme environments. And for me, I am not existentially threatened by things like cancer or obesity or even age. That's an individual object. We're existentially threatened by the change in our planet and the fact that our resources are decreasing, that our diversity is being destroyed, and we need ways for fending against it. And this was not a way for fending necessarily, but it was a way of thinking about manufacturing and resource supply in a way that was different than other people were thinking about it. And I like that idea. This other one is a little bit more frightening and less aspirational. And this is a graph from the USGS. And it shows where we, in the United States, have contaminated the land with uranium and nitrate. And it's not actually that we put the uranium there. We're a uranium-rich nation. It's part of our bedrock. And what you'll see, this is where I live, around here. This is the California agricultural belt. And that you see all is where we use the Haber-Bosch process to feed the plants nitrate fertilizers. This has become poisonous. It's where we've poisoned the groundwater and we're beginning to destroy the soil. And I'm very interested in how do you restore that land? Now, that looks bad enough, but this thing is absolutely horrifying. This is the Agua Agua Aquifer. This is the major catchment of water in the United States. It's how the rain and runoff from the mountains is collected naturally. And we pump that water to feed our breadbasket, our crops. When we pump that water, we suck the nitrate into the groundwater, into the table, and that's now it's nitrate contaminated. Nitrate in water makes nitric acid. Nitric acid dissolves rock. Dissolved rock makes minerals come up, in this case, a lot of uranium. Now we've overpumped this well. We've pumped it so it's low enough that we've concentrated this to extremely high amounts. If we pump any more, no more water. We won't be able to feed ourselves. If we pump, if we've gone below the levels we can restore it in reasonable time. Kansas farmers are now pulling up uranium in their corn. So we're at a point now where we have to be able to remediate these lands, recover these lands, reduce the water usage, reduce the runoff. There's lots of follow-on effects from this. Now, all that said, you think, well, good, you live in California, work here, this is the major existential threat to our breadbasket, work there. No, because money is where the money is. And so instead, I ended up working there. Now, that's not that's not as interesting. It's interesting historically because this is Oak Ridge, Tennessee, and this is a classified site called Y12. Nice science fiction-y thing. And it's where we extracted uranium to make nuclear weapons. And it's a watershed with a river running through it. And it's highly controlled, which gives me the ability to operate down there. And I want to talk about this just because when you work at a site like this with such a high amount of control, where you know exactly where the sources of pollution are, where there are point sources. In this case, there's a point source right here in something called the S3 ponds, where we buried all this material. This S3 pond is now a parking lot, and cars run on top of this parking lot, and they pump on this containment layer, and it forces the uranium and nitrate out, and it forms this gradient down into this creek. And we can study this little area called Area 3, where the plume is built, for example. So I want to study this in great detail. And I'm doing this because I want to get this model. I want to get this model that Ahmad, for example, to talk about how do you build these models. What are you going to do it? And whoa, there we go. And we go out there and we measure in excruciating detail the sediment and the water and how things move and what they do. And all these little columns here you see are these wells that we drill so we can see what's going on in there. And then we can measure in time and in space things like all these different things, pH and metals and carbon and uranium. And we can measure where the organisms are. And here you can see that this is depth and this is an organism, and they're stratified into different communities that organize themselves in space, and this is natural. I didn't do anything here. This is what they do naturally. And then I can ask, what genomes are there and how those genomes were arrayed in various ways. And to me, this is the context I was after. So the idea was once I have this, I can begin to put them into mathematical models. And this is just for you, Ahmad. So this is the reactive transport model in which we measure how the abundance of chemicals and organisms as they move in space and in time, based on their ability to flow in the water, to attach and detach from mineral surfaces, to grow on the substrates that are present, to interact with each other, and to do so based on their genomes, all in one place. And this is where the engineering begins, because now I can put all that context into this mathematics. And I can ask things like, well, who's there in the environment? And so when we extract these organisms, this is an example where we found these organisms, and these organisms are living there. And inside of them, there are these things called talosins, for example. These talosins are ways that they interact with each other. They kill each other using them. They're like little tiny phage, but they're not phage, they're phage bodies. And then I can go all the way down into individual genes. And these are all the little genes. These genes are found in the environment, in organisms that I can get into the laboratory and understand. And I build methodologies with my partner, Sir Adam Deutschbauer and Vivek Metallic. We build methodologies to query every gene in the organism. So we use overexpression and underexpression and repression to do this at genome scale. And we can discover how every gene plays a role in this organism to survive in these environments. Now we have these maps, gene to organism, organism to environment, gene to organism to environment survival, or growth, or whatever. And now we're bringing them together into those models we just told you about. And to do that, we use this thing called KBase, which Ty mentioned. And then this is the big open source system that I developed. It's developed in open science. Has about 405. Well, actually, it's about 460 open source tools now that go from reads processing all the way to full models of communities. The whole community is working on this. We have about 150 community developers who work on various things in the system, from protein structure to 3D metabolite measurements and things like this. And they build these things called narratives, which are basically things you can do online, which are like Jupyter notebooks, where you can build papers in place with your data and your tools and your analysis. And it's all provenance and set up so that I can do machine learning on it when we're done. Right now, we have about 70K narratives in the system. People have built, people read it from all over the world. And this is what I wanted to get to this for this group. We have people begin to use this for education. We've got 170 educators worldwide who've begun to use this system to teach how to do this integrative data analysis for predictive systems biology. And can get the types of models I told you now, starting from reads and concentration measurements and growth measurements. And this is sort of the workflows that they teach in this course. You can go there and use those narratives as teaching materials right now. And this has been used, all over the world to make these things, to begin to get to describe these things. And what I like about this system is this is, for example, a professor who works in South Africa. She has, she has driven her postdocs and students which are mostly in classes. You have 70 accounts that made 25 K-based pubs totally for free. They use DOE compute and all our tools and everything for free. It's totally open access. And from that, they've been able to get 25 publications from her course. So it's pretty cool. So now that we have these systems operating, we're now getting back into design mode. We now have this idea that we can measure things in the world. We can begin to model them. We can put them together. This is just an example. Vivek and I have been thinking about how to put together this thing called a phage foundry. We found many phage in the environment, many telosins in the environment. Can we begin to engineer them and reorient them so we can edit communities and treat resistant infection and things like that. And this is where we get back to the scale that I operate on, where we put these things together this way. Okay, so I've taken a lot of time. I'll just stop by saying that, look, doing this is me finding people to work with. And before everyone got here, I were talking about how we find people. And that's one of the things that, despite the fact that I am generally an introvert and like to work by myself, I found necessary to do. These are the folk I work with who make all this possible. And people are hard and it's difficult, but that's how we get things done. We are a people working together. And if we're going to understand context and we're going to get things accomplished, we need geneticists and theorists and synbiologists and metrologists and chemists. And you only do that when we have groups this large. You can't do it in one brain. So with that, I'll stop and give over the floor.
Wonderful. So now I understand completely your transition from SynBYSS era to the last 10 years, because I kind of wonder, because based on your kind of paper kind of completely changed from the before and after SynBYSS. And then now I completely understand what you're talking about. And that's amazing, because this is a very important problem, because I also believe planet health is more important than any other thing at this moment, because we have the lots of problems currently we're facing. So that's absolutely wonderful. And also, you know what, I actually already submit a paper about governing principle in biology. And you kind of mentioned some of that I already included in my governing principle. And then we'll see, whether that pan out well with the big journals. So thank you again. That's wonderful. This is amazing. I love it.
Thank you. My pleasure.
Yep. Okay. So now, the main speaker of today with a longer introduction, Dr. Ahmad Mannan is trained as a theorist, but has had the pleasure of working closely with experimental biologists throughout his research career. He used a mathematical and computational approach to elucidate the design principle of genetic circuits that control cell metabolism, and to designing synthetic control circuits for developing efficient microbial cell factories. He did his undergraduate and master's in mathematics at Imperial College London, and PhD in microbial systems biology at the University of Surrey, UK, where he developed kinetic models of the allostatic control of the E. coli central carbon metabolism. It was his subsequent postdocs in the group of control theorists and collaboration with experimental synthetic biologists where his love for synthetic biology really took off. During his postdoc at the Department of Math at Imperial College London, he worked on elucidating the design of the genetic control of fatty acid metabolism. In the Warwick Integrative Synthetic Biology Center, he also developed computational tools to rewire cell metabolism and designed a irreversible metabolic switch for controlling efficient microbial cell factories, more of which he will talk probably today, I guess. And recently he was a postdoc at Imperial College, and now I believe he is leading his own group in designing strains and microbiomes for treating acute bacterial infections. Ahmad is a true rising star with huge potential, and I look forward to listening to his talk today. And Ahmad, thanks so much for your time today, and please take it away. And it's all yours now. Thank you.
Thank you so much, Tae. It's such an honor, such a pleasure, such a nice introduction. And thank you so much, Adam, for your amazing overview of context dependency and the importance of context dependency in biology as well. I'm going to try and share my screen. Let's see if I can do it. Okay, just let me know when you see it and if everything works out.
Now working. Thank you.
Okay, perfect, perfect. Yeah, thank you so much. It gives me such a great pleasure to be on this webinar and seminar series. And my name is Ahmad Mannan. I'm a postdoc at the moment at the Department of Bioengineering at Imperial. And today it gives me a great pleasure to be able to talk to you a little bit about computational designing of bacterial cell factories for efficient chemical productions. Let's see. There. Good. So bacteria, everything's going to be focused on bacteria here. Bacteria can be harnessed as microfactories for the synthesis of a wide variety of useful and high-value chemicals from cheap feedstock and even from chemical waste. So we want to produce, we want these bacteria to produce chemicals, and we want that production to be efficient. That is to have maximum volumetric productivity. There's a maximum amount of titers that we get in a minimum amount of time, and convert as much of that input substrate to an output product. So we want to maximize on yields as well. We want that production to be robust. We want to be robust to the heterogeneous bioreactor environment and to evolution or whatever environment that it sits in. But since we're asking a live bacteria to synthesize chemicals that it naturally doesn't, or in high quantities, then there's a fundamental trade-off with cell growth. High synthesis and low growth may mean that production, you get good amount of production, but only from few cells. And low synthesis, but at high growths may mean that you get low production from large populations. And there's probably a sweet spot in between where you have a maximum amount of total product synthesized. And we can take two core strategies to deal with this trade-off. We can try and create an optimal balance between growth and production. And growth and production can happen hand in hand, a strategy which we call static design. Alternatively, we can copy quite a ubiquitous strategy in nature, where cells can first grow maximally, and then we can add an inducer and get high populations to produce the product. Switch all of these cells to production mode, but that comes at a cost to growth, a strategy which we call dynamic control. So to help us to figure out how to design the production phenotype, that is the static design strategy that I was just mentioning, we asked a bunch of questions. Like, what is actually that growth synthesis trade-off? What does it look like? Where in the metabolic map of, the genome scale metabolic map of metabolism of a cell, where should we intervene and help to redirect that flow of the metabolism to create the ideal balance between that growth and production and to achieve that maximum volumetric productivities and yields? Also, can we achieve a production that is robust to evolution, robust to perturbations? So to uncover these growth production trade-offs, we use models, mathematical models, known as genome scale metabolic models. So I just want to go into a little bit of detail about what those models are, so it just sets the scene. In brief, these models are like a system of ordinary differential equations with the rate of change of metabolites, concentrations, as a sum of all the reactions that are making it and consuming it, weighted by the stoichiometry of the chemical reaction that uses or produces it. We don't know what these reaction rates are in terms of the metabolites. We might not have that knowledge, especially at a genome scale. But we can evaluate that system at a steady state. So what we're left with is solving a system with coupled linear equations, linear in the reaction fluxes themselves. And since usually in metabolism, there are more reactions than metabolites, then there are many more unknowns, that is reactions, in these equations. And we don't have enough equations to solve for them exactly. So this is converted into a linear programming problem, where we're maximizing one key reaction, maybe a reaction which is representative of the growth of the cell, given some constraints of the chemistry itself that's happening in the cell and thermodynamic bounds on the reaction rates. So when we solve this problem and look for the ranges of possible production values for a given growth rate, we actually determine this sort of relationship, which is the picture actually of the growth production trade-off that we wanted. We can also go back and solve this linear programming problem with certain interventions, where these interventions can be knockouts of certain reactions as equivalent to like knocking out sets of genes to knock out those reactions. Or it can be the up or down, simulating up or down regulations of these reactions by constraining sort of the flux values that these reactions can take. And for certain interventions, we might find a trade-off that looks a lot more like this. So what's that? This part in red is actually known as growth coupled production. And looking for knockouts that can enable growth coupled production has some really key advantages. There is here a non-zero minimum guarantee production flux, which is maximum at that maximum growth rate. And once the knockouts are implemented in the lab, the key problem is that cells don't usually perform at this maximum straight away. They usually perform somewhere, their performance lies somewhere over here. And it is really, really suboptimal. And that's because we've messed up, we've really unbalanced all of metabolism because we've intervened and messed up their traffic flow. But however, studies from the lab of Bernard Powelson from University of California, San Diego have shown that adaptive laboratory evolution as a method in the lab has allowed for strains or mutants to converge to the optimal flux distributions. And on the back of that, the study of Tokiyama et al. From Osaka University in Japan showed that adaptive laboratory evolution of a strain of E. coli with knockouts that could enable the growth coupled production of succinate started off poor, but after adaptive laboratory evolution, its performance was close to the maximum predicted. So can growth coupled production be the way to go? But how applicable is this? How widely applicable is this? Can this be done for any chemical product of interest? The study of Klampt et al. From Max Planck University in Germany showed that you can find growth coupled designs for almost all host metabolites. And a host metabolite is going to be the precursor for any product that you can put into it. So this is quite promising. And not only that, but it can be true for the application of several microbial hosts as well. So this seems to be a widely applicable strategy. Also, if we evolve to that maximum performance to get that genotype, then the production coming from that genotype is robust. It's probably robust to evolution as well. So this got us thinking, especially that last point about robustness to evolution, that what if we could find knockouts that could give a production envelope that looks instead something like this, where the strain production could actually become very robust. Here, if there is any impediments in the synthesis to a synthesis of the product because of mutations that are happening during evolution, and that would, mess up the optimal flux to that production, then this would strongly reduce growth and so impose a strong selection pressure for the high producers. Conversely, any fluctuations in the growths that are caused maybe by a heterogeneous growth environment in large bioreactors or wherever it's growing, this can appear to actually only cause a small loss in production, that making production robust to the growth fluctuations as well. And this is what we define as a strong growth coupling. So how do we find where in the metabolic map to implement the knockouts to create strong growth coupling? But we also want to maximize growth and product synthesis. And why only knockouts? Why not up and down regulations? Well, if we're going to harness adaptive biology evolution in the end to evolve to the maximum performance, up and down regulations can be easily retuned during that adaptive biology evolution. But knockouts are really hard to bring back depending on how you make the knockouts. So we posed looking for knockouts and this as a multi-objective optimization problem. And together with Lawrence Legon of the University of Warwick, who did all the hard coding work, we developed and published a MATLAB-based tool to solve this, which we called GC-Front. There are a few other computational tools in literature that look for growth-coupled production designs, but the advantages of GC-Front was that it could output many Pareto-optimal designs of varying coupling strengths, growth rates, and synthesis rates, and this vast array of design choices gives the user the flexibility to select designs based on the best trade-off between growth and synthesis. Evaluating coupled strengths simply from clicking on a given design, because it's actually an interactive window when you use GC-Front, and even output all the metrics of the designs in an Excel table. You can do further analysis like calculate volumetric productivity and yields for each of the designs, rank them to evaluate if it's worth actually trading off growth and production for the coupling strength, because it may not be worth it, and even do an econometric analysis based on the cost of the substrate and the value of the product itself to select which of those designs you can take forward. So GC-Front can enable us to determine what genes in the host to knock out and redistribute the flux. But we should actually also consider whether we need to overexpress the enzymes of the product synthesis pathway too. So far, we only know how to redistribute flux towards that pathway, but not so much what's happening there. So in effect, this could enable us to further open up the valve at that production that we're redirecting the flux to. Also, how does that overexpression affect the production itself? So there are limited resources in the cell. And what I mean by that is the energy is made from consuming and metabolizing substrates and limited amount of ribosomes. And in the cell, these are used and distributed very carefully, orchestrated to meet the demands of everything that the cell needs in the right proportions to enable it to grow. So overexpressing enzymes of the synthesis pathway itself will sequester the ribosomes and drain the energy, but it's unclear how this actually feeds back to affect the production itself. So in collaboration with my good friend Alex Darlington from the University of Warwick, who just spoke a couple of weeks ago, we sought to address this question using a simple self-replicator model from Alex's previous works. So in brief, this models a nutrient-dependent production of energy, which drives ribosome and energy-dependent synthesis of enzymes and ribosomes, and can model growth based on total cellular translations. So we added a product synthesis pathway and associated enzymes that drains a host metabolite. And from tuning its translation rate to emulate increasing expression of the synthesis pathway, we actually saw a deleterious effect on growth. So increasing translation, growth rate went down. It was more deleterious when it wasn't just the expression of the enzyme, but we drained metabolism as well. So this is, we're trying to figure out like, why is that happening? And it's not very surprising to find that actually it's because the higher the expression, the more ribosomes were sequestered, and then overall this drove down the amount of ribosomes that the cell was able to produce. However, what was surprising was that with this decrease in ribosomes, there was a massive shift in the whole proteome profile of the cell. It was actually a relative increase in the cell's proteome fraction that is all the metabolic enzymes, the transporter, the host enzyme, or the production enzyme. And this in turn increased production until, of course, you get too much overexpression, and then this causes burden. But it's because of the increase in transporter enzymes that you got an increase in the input, and then you got an increase in production as well. So in tandem with redistributing flux to that production synthesis pathway, we should perhaps look for creating a little bit of burden, an optimal amount that would create actually further production capabilities of the cell. And this is all driven by a natural feedback, which is created from limited ribosomes and limited resources of the cell itself. So designing production strains using the static design approach is great, but is restricted to a fundamental constraint of that trade-off between growth and production, because you're trying to get that right balance. So however, a more promising alternative strategy is to design production strains that implement production over 2 stages, growth and production. So you're decoupling them. As a growth stage, we grow maximally, and then you induce a switch, and you harness an inducible genetic circuit to be able to make that switch. It does come at a cost of growth, but if there are enough cell factories, then it's probably worth that cost. Of course, this is a copy of a ubiquitous strategy in nature, where there's a lot of transcription factors and other genetic circuits for metabolic adaptations, such as the lac operon coron sensing systems that produce antibiotics in microbial communities. And there have been several papers, many papers actually, showcasing the dynamic control strategy and the implementation of it experimentally from the groups of Christian Madhavan from Toronto, Kristalla Prather in MIT, Tazo Hanai in Japan, and Yujin Yan as well in the States. They all seem to show one thing, that there is a promise of higher production capabilities because you're uncoupling that growth and production. And this is after tuning a little bit that genetic circuit, but mainly finding the optimal time in which to reduce these cells. But there remains a key problem, and that is the cost of adding the inducer in the first place. And this can really severely limit the scalability of the strategy to large-scale bioreactors in production. So we could use cheap nutrients instead to induce that switch. There are many examples of nutrient-inducible systems in nature. We can harness those. But the main question remained is to understand how to design these inducible circuits so that we could minimize that total amount of inducer use, even if it was a cheap metabolite, and how we can keep that production on as well. So to understand how to engineer an inducible genetic circuit to address these questions, we studied the fatty acid inducible circuit from E. coli as an exemplary system, a system which is actually native not just to E. coli, but to many bacteria that can utilize and take advantage of fatty acids from the environment. We developed a mathematical model of this system based on our previous collaborative work with the lab of Fuzhong Zhang of Washington University in St. Louis on understanding fatty acid inducible control in E. coli. And we extended that model so that the main regulator, the transcription factor FAT-R itself, could maintain growth in the absence of fatty acid, but then switch to production. It doesn't matter what the production is of, but when the inducer, when oleic acid is added, the fatty acid is added, it switches. So from simulations of adding that inducer with the aim of how do we add it so that we can keep it at production phenotype, we found that the system actually requires long periods of induction and recovers back to switching off production very quickly. And that's not really a good combination of behaviors if we want to address these questions. So what is driving this quick recovery in the first place? When induced with oleic acid, the internalized nutrient binds to and sequesters the FAT-R, and FAT-R is actually negatively autoregulating the cell. So this releases its self-repression, which makes more of it, it gets bound, and then you get an accumulation and storage of the total amount of FAT-R, which when you run out of nutrients is quickly released and then quickly re-represses production. But we found that replacing this negative autoregulation here with a positive autoregulation instead fundamentally changes the dynamic behavior of the system now. We don't get that accumulation. In fact, we get a dilution over time of this transcription factor storage, because inducing reduces its self-induction, which means it can't produce more of itself, and then that initial storage is diluted away as cells grow and divide. Now, when the inducer is stopped, less FAD-R is released, and since this cannot activate its own expression well, it will take FAD-R time to build up, so that's why there is this delay in its accumulation and ability to switch off production, and this drives slower reversions in the end. But also, low FAD-R abundances mean less to sequester, which is why we see shorter reinductions as well. Now, tuning for different rates of how we feed in that inducer in the first place, we find that overall, the total amount of inducer used, which is on the x-axis here, in the circuit with positive autoregulation, that is all the blue, and the area under the curve is the total amount of inducer used, to be far less in circuits with positive autoregulation as compared to negative autoregulation. But when we do a mathematical analysis of this motif, the circuit motif, we can actually mathematically prove that the circuit always reverts when you run out of the nutrient. It just it's a reversible switch. It can never stay stuck there. But in this dynamics, we saw an opportunity from this analysis of the system's dynamics. And in the circuit with the positive autoregulation, when FAD-R expression goes down, we asked, how could we actually keep it locked at those low levels? So to do this, we added this toggle-like circuitry so that during induction, another transcription factor, such as TET-R, can suppress FAD-R expression and block it from activating itself. Now, when that happens, only a short temporal addition of inducer is needed to switch the production phenotype. And if enough TET-R is expressed, then it keeps it locked there, even when the inducer has run out and depleted because it's being metabolized by the cell itself. And now that we only require a short temporal induction, there's a drastic reduction in total inducer use. And so very minimal induction costs. And it doesn't matter how long production goes on for, and this will be especially great for feed batch cultures if you want to continue on the production phase. So we should look to engineering an irreversible switch in the context of chemical production in bioreactors, maybe not in nature, of course. However, just this circuit motif is actually not sufficient to allow that irreversible behavior. So we did a parameter exploration and how that affects the irreversible behavior itself, and extracted some really simple design rules to push the behavior of this irreversible switch to push the behavior of the switch into being irreversible in the first place. And in particular, we found from the analysis that the model predicts that we should engineer for strong expressions of both of those transcription factors, but where the inhibition of each other's expression should be relatively weak so that it doesn't just stay stuck in the on or off stage. We found that we want to minimize the total amount of inducer used. We can expose for very short periods of time, but at the same time at relative high feed-in rates of that inducer itself. And we know that selecting the right time to induce the switch to production affects the total productivity of the culture. But what was really nice to find is that we can actually tune that switch time used by just tuning the basal expression of the enzyme which actually takes up that inducer in the first place. We can tune the basal expression to change the switch time, but it has almost no effect on how we tune for minimizing the total amount of inducer use. So in effect, we should be able to tune switch time and inducer use independently. And that gives us a lot of flexibility in engineering this irreversible switch, to which addresses our original question. So what are the implications for an irreversible switch? Well, it saves induction costs and makes inducible production more scalable. But what could it be doing? Could it be doing something to increase robustness as well? The cellular production has been shown to be quite heterogeneous, even in small scale productions with around almost 50% of cells in this paper showing sub-levels of production. We speculate that the irreversible switch may enable production to be robust in heterogeneous and fluctuating levels of inducer availability in large barrier. So if we added the inducer, there could be fluctuations at level of inducer and whether the cells switch on or off. But once they're switched on, it should stay there. So as long as the dynamic range of the switch is large enough so that it's hard for stochastic gene expression to flip the switch back, it could help to create robust production. So now that we have a design for the switch that can enable robust production, there still remains a question as to how to fine-tune the switch to design to achieve maximum production, maximum volumetric productivity and yields. And that switch, what should it control? So in the dynamic control strategy, there is a massive shift in metabolism from growth to production, which means that there is a big shift in the redistribution of cellular resources. And to uncover how to engineer a dynamic control switch in the context of a cell with limited resources, I went back to Alex and we collaborated on, we continued our collaboration and we developed a model where the switch is essentially a black box, but considered 3 core motifs of the dynamic controller, where the switch can control the expression of the host enzymes, or the production pathway, or both. And using this model, we defined a multi-objective optimization problem to find the optimal regulation strengths, the expression rates, the reduction time that will maximize both volumetric productivity and yield. And when we solved that optimization problem, we found a Pareto front of optimal designs along these Pareto fronts. We first found that actually shorter induction times could increase yields, but that comes at a cost of volumetric productivity. But just thinking back to the designs, intuitively, when designing a control switch, you would think that controlling only the expression of the synthesis pathway enzyme would be a good idea. However, it seems that selecting this motif gives you actually quite suboptimal performance. And instead here, we see that the other motif, where we're only controlling the expression of the host pathway and the host enzymes to shift that redistribution into the production pathway, seems to give near optimal performance, suggesting that perhaps we don't need to control both the synthesis pathway enzymes and the host, but this does come at a cost, that we do need to have strong expression rates of that host enzyme, and we need to have strong control over it. And this can become really difficult to engineer in the lab. Having control on both the host enzyme and the production enzyme, actually, we need there's a lot less of a demand. We need a lower expression rate and a lower level of control, strength of control on it. And this is much easier to engineer in the lab. So perhaps the way to go is to engineer to have that dual control. Right. So we've looked at how to design production strains through static design, dynamic control, but it was still unclear to us which strategy will enable the maximum production capabilities of a given host, producing a given chemical in a given defined media. Also, in the dynamic control strategy, it was still not clear which genes of the host cell metabolism should be placed under the control of the switch. So in order to address both these questions, we developed GC Front 2. It uses the genome scale model of the host and explores sets of genes to turn off or even to knock out, assimilates batch culture production, and solves a multi-objective optimization to maximize volumetric productivities and yields by exploring over which genes are turned off and which ones to knock out and when to induce the switch, and outputs designs and their performances. And as an example, just as a teaser, because this is just still in preparation, we looked at the design for the maximum production of 4-deoxygadosol from E. coli in M9 minimal media. And we found that designs that were through dynamic control could give an improved production capability. So I believe that this tool will allow one to explore different growth conditions, different hosts, different production strategies. And this really opens up the design space to find the highest production capabilities of a given chemical of interest. So some of the key take-home messages I'd just like to summarize is that from static designing, GC Front as a tool can find knockouts for growth coupled production. Why do we want to look for growth coupled production? Because we can harness adaptive biology evolution to evolve strains to maximum production capabilities, and where those production capabilities are robust to evolution and to growth fluctuations. And of course, we can then, in tandem to that, tune up production, the production enzyme expression itself, because a light amount of burden to the cell can actually redistribute the proteome profile of the cell to a relative increase of the amount of enzymes, and this can actually improve production even further. What about dynamic control? An irreversible switch can make inducible production much, much more scalable. And having dual control, that is having control on both the host enzyme that redistributes the flux towards a production and the product synthesis enzymes, can actually make it easier to implement and to design this control in the lab. And then putting that together in one single framework, GC Front 2 can help, as a tool, can help to identify which genes to knock out, switch off, and which design strategy can open up that capabilities of the cells. Now, following from this last point, I just wanted to share my opinion on how these computational tools could be embedded into what we envision as an automated biofoundry for the manufacturing of cell factories for specifically for chemical production. And what we could have, this is just a brainstorm, but what we could have is design from GC Front, which churns out the gene sets to target, to turn off, or to knock out, and gives us the prediction of the performance. We could get robots to build these in a lab. This is already kind of happening. I'm not saying anything new. With you know, if static design is the way to go, then it can implement those knockouts and evolve them in an automated manner. If dynamic control is the way to go, then you can add the reversible switch. You can create libraries so that it's effectively tuning the parameters of these strains and then test them, doing benchtop batch culturing and comparing with the prediction. And if there are problems that there's massively off prediction, then identifying the bottlenecks will have to be done more manually through a learning process. This can either feed back into parameter tuning, creating mutant libraries, or a complete redesign. So there are biofoundries popping up all over the world, and it's growing. So could these tools be useful in that respect? I also just wanted to finish off and share some really exciting stuff that I'm working on now, which is a shift away from the biotechnology area that I've been talking about through this entire talk and the tools developed for that, but the same mathematical and computational tools to be able to figure out how to produce chemicals and how to redistribute a flux and make chemical production from these strains robust, can be applied to create live therapeutics. And currently I'm working with a great bunch of people on developing the live therapeutics for eczema. Just a little bit of insight. So eczema is a skin inflammation. It's quite rampant. So about 20% of all adults and 3% of children have eczema in the world. And this is growing. But this could cause dysbiosis of the skin microbiome as well. And people with eczema get Staph aureus infections all the time. So what we want to do is we want to create we want to engineer a skin microbe to sense skin inflammation and deliver a therapy according to that. And I'm really looking forward to working with all of these amazing people understanding the skin microbiome, how to develop the circuitry to engineer for the live therapy, and how that all fits together in the end. Thank you very much.
Wonderful. So that amazing talk. I also love to see all the colleagues in European country, including Mark and Alex, also working with you. That's amazing. So Adam, do you have any question or comment?
Well, I think my main comment is I think the approach is really very interesting. I think the fundamental idea that you can use this optimization framework for driving experiments automatically, I think is one of the critical capabilities we have to develop, I think, across scales. Even
Right.
for things, even as things as open as your eczema experiment, ultimately being able to monitor what's going on and then intervene so that you can get the effect that you want is going to be part of this. Now you're talking about in the manufacturing, something which is requires genetics in between. You don't always need genetics, but when you do need genetics, I think this fundamental idea that you're trading against fitness and activity is a critical insight, and I think that it's one that we over the past decade and a half we've seen a lot of maturity in, but linking it out to where it becomes like where it goes over large periods of time, like when you're doing like months in a reactor or months in a human being and they're under selection is going to be extremely interesting. But I think it's a very cool approach.
Yeah, no,
So
thanks so much. Yeah.
Thank you. I have one quick question before seeing any question from audience. So in 2006, my PhD proposal is all about dynamic control. At the time, nobody talking about dynamic control, and I couldn't implement that one because I need to construct my own pathway to implement the. And then Chris Predator actually assigned another student to finish that project, and then later on we are working on that project, except me. But the question I have is, I love dynamic control, but in terms of industry application, the all current dynamic control or prediction on model focus on small scale modeling. And then even your diagram also show those cycles is stay in the small scale and then scale up later. But I believe to be practical, the control and prediction model should consider scale up situation, and then that should be done. Otherwise, I mean, I would rather go with the traditional strain without dynamic control and then scale up and then produce, and that's typically more production based on my experience. Could you comment on that?
Yeah, no. So scale up is a fundamental question. Doing all of these experiments on a benchtop reactor is probably fine just for the proof of concept that you're actually producing something, is good. But scale up, there's a lot more problems associated with that. So one of those, I think, is one of those key problems is scale up is people are scale up, they're a lot more worried about, from my understanding, they're a lot more worried about the amount of nutrient and feed in the feedstock itself, what the costs associated with that are, how that trade-offs with the amount of product that's actually produced in the end. So there's a lot more economic costings for that. Also, robustness of strain and whether the strain's evolutionary robust or not. This is a big question. So, alluded to the fact that looking at growth coupled designs is probably the way to go if we want to have evolutionary robustness, because then if we're going to be doing these going to be costing for production in huge bioreactors and over long periods of time, then we've got to ensure that production itself and the strain itself is evolutionarily robust. So this could be the way to go, but it's yet to be tested. Bigger, I think, when I spoke to people about upscaling and what are the key kind of issues with upscaling, what do they consider as the key issues of upscaling? They said it was more the downstream costs, the purification steps, the taking out the product, which is more of concern. That's where most of their costs lie. So everything that I was talking about was really at the upper end of production, not so much the scale up itself.
Mhm.
So sorry, I don't know if that really answers your question.
Yeah, I mean, there is no the correct answer for that question because that's an
Yeah.
open-ended question, and then that
Yeah.
that should be done by you and other people, and also experimental people, because they do not consider that important fact, and then many people, so even industry do not consider dynamic control at all because that is not practical solution for right now. So in the future, you should have implement those kind of consideration in that way more practical for the real industry people. That's my point.
Yeah. I guess the dynamic control is it's very limited right now because of induced costs. That's why I was focusing during my postdoc in Warwick. That's why I brought up that question in the first place, because everybody I talked to about production from cells, they say, oh yeah, dynamic control sounds great, but you have to add an inducer. Why would you do that? That's an added cost, right? Could you capitalize on something else? Could you capitalize on what nature is already giving you, such as quorum sensing, right?
Yeah.
So yeah, no, absolutely. But then
to me, to be clear, I mean, if somebody add the inducer, I do not call it the dynamic control. The dynamic control should be without added inducer.
Yeah.
That's my original idea.
Yeah, exactly. But the irreversible switch that I'm talking about, it's irrespective. So you could look at it from two ways. So one is, you can add an inducer externally, and companies themselves, when we had a chat with them, they want to go something really, really simple. If we said, okay, look, we have an irreversible switch that can be automatically turned on when it reaches a higher cell density count, they're like, oh, that sounds too complicated.
Oh, we lost him.
We can't add it into our process, right? We want to use current established to do it. So current processes are, they already have bioreactors where they can add stuff into it, and they say, oh yeah, we can add an inducer. Do you have a solution with that? But then there's induction costs which offset that. But the main point isn't so much the cost of induction, but more that can induction stay on? Because the inducer itself, if it gets metabolized or if it gets or if it breaks down or whatever happens to it, induction will just not stay on for long enough. So, and there can be fluctuations in the environment as well. So maybe an irreversible switch could help in creating robustness in keeping on that production stage in the first place. So it doesn't matter what the induction is.
I see. All right. Adam, do you have any other thing to add or comment, question?
I think that one of the things I put here is that we often think about this type of control in terms of scale-up systems, and I think there's a real, obviously that's a huge industry and we have to respect it. But I think there are other types of industry where the dynamic control, especially the adaptive control you're talking about, Ty, where it's internal, where you're sensing and adapting yourself to the environment. That's where applications that are not in big reactors, that are about production of commodity chemicals that are a cent a gallon. We're talking about things that are living in your gut, or which are living on your skin, or living at the root of a plant. They kind of have to have those systems. And my sense is that we're learning how to do that here. Right. And much more is from the point of view of containment, having to add things to the system are important. Open systems. So having something that is an inducer that goes away actually ends up being important because it contains your system. Is where it is. So you can change your viewpoint to say the application isn't a 10,000 liter scale up reactor. It's a bioreactor for a crop, or it's a probiotic for gut health or skin health or lung health. I think that's an important distinction here.
That is exactly what I want to say. I mean, thank you. I mean, just to elaborate, what I want to say. That's absolutely right. Okay, so let me check whether we have question from audience. And then this is already the central time 11:17, so probably we need to close. Okay, so thank all for joining and staying today. We'll meet again on July 13th, Thursday, the same time, the same Zoom link. We'll have Professor Bamsi Musa at Harvard Medical School, Howard Hughes Medical Institute, and TL To from the same laboratory. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I'm stop recording. Just give me 1 second.